Hi and Assalamualaikum. My name is Nurul Ain binti Muhammad Liki. My number matric 23-20F1012. Today I will do lab simulation for bacteria ID lab. For this lab have 6 steps, which is sample prep, PCR amplification, PCR purification, squeezing prep, DNA squeezing, and lastly squeezing analysis. First of all, we have to click the glove to open the drawer. After that, we'll obtain a sample from the culture disk. Click the wire loop to continue. Click on the bacteria colony in the disk and then click the microcentrifuge tube to transfer the bacteria colony from the wire loop into the microcentrifuge tube. Placing bacteria colony in this tube. Now we already add this captive enzyme to the sample and then click yellow micropipette to the continue. Adding the digestive bubble to the microcentrifuge tube and discharge the tip. After that, the microcentrifuge must sit for several hours. Waiting several hours. After several hours of enzymatic digestion, your sample is ready. Moving the microcentrifuge tube into the water bed. After that, the digestive enzymes are now denatured and inactivated. Now you will spin down cellular debris for removal from your sample. Centrifuge is complete. Unwetted debris have now been spun down. The DNA we want is in the supernatant. Click the microcentrifuge tube to continue. Transferring the supernatant to the PCR tube. The sample preparation is complete. After that, we will do step 2, which is PCR amplification. First, we will add the PCR master mix solution to all tubes. After that, we need to set up the positive control reaction. Click this. Adding control DNA to the positive control PCR reaction tube. For the negative control reaction, we will add water. Adding water to the negative control PCR reaction tube. After that, we will have to load the PCR tube into the PCR machine. After the PCR tube have successfully been loaded into the PCR machine, Click the PCR machine to continue. The PCR has already running application. This is the result. Done. After that, we will do the step 3, which is PCR purification. First of all, we should set up the microconcentrator column. To do that, we should click this yellow bottle and then adding buffer solution to the column. Click the orange tray to continue. Adding PCR product to the column. Next, the tube containing the PCR sample, positive control and negative control, will be put on ice. After that, to tray the column into centrifuge. Edit the counterbalance to the microcentrifuge and close it. Waiting 50 minutes. DNA is now trapped in the column. Now we need to loosen it into a new tip. Adding a new tube. Click the inverted column to continue. Adding buffer solution to the inverted column and discharge the tip. The first collection tube is no longer needed, so we should discharging the first collection tube. Now you will centrifuge the assembly to collect the DNA into the collection tube. Edit the counterbalance. Click the centrifuge to continue. DNA has now been collected in the collection tube. The column is no longer needed and will be discharged. The PCR produced has been purified. Now we will do the step 4, which is squeezing prep. First of all, we should dilute the PCR product to increase the volume. Adding distilled water to the purifier PCR. The green and blue strip tube already contain PCR reaction mixture in them. Adding purifier PCR product to the strip tube. After done, we have click the PCR main stain to the continue. The strip tube have been loaded. Running PCR amplification. Click above to pause or reply the animation. This is the result for PCR amplification. The squeezing preparation is complete. We will do part 5, which is DNA squeezing. Load the automatic squeezer. Click the DNA squeezing machine to open the doors and load the tray. After that, click the orange tray to load it into the DNA squeezing machine. After that, click the inner gray door above the orange tray to view the DNA squeezing animation. Now squeezing. We have to wait to see the result. Done. We are already complete the DNA squeezing section. We will do the step 6, which is the last step, squeezing analysis. This is the bacteria Bartonella hesseli.